Today is April 23rd, 2013. My name is Tracy Dodd, and I'm in the Boston Convention Center during our annual American Physiological Society meeting. Today I have the pleasure of interviewing Dr. James Bassingthwaighte for the Society's Living History Project. Dr. Bassingthwaighte is a long-standing member of the American Physiological Society, and his research focuses on cardiovascular physiology. Dr. Bassingthwaighte, welcome.
Well, I was born and brought up in Toronto and Belleville and Ottawa, Ontario. My father started as a lawyer, but became a businessman in radio. And my mother was the daughter of we crossed the family crossed the border generation after generation. So my grandmother was a Canadian. My mother was born in Indian Territory in what was to become Oklahoma. She was born in 1905, and Oklahoma came in 1910. But she was musically inclined. Her father was a U.S. consul around the world, and she grew up mostly in France before and during the First World War, and so she was brought up in a pretty, fairly cultured kind of enterprise, and she and her sisters gave concerts and things like that. And my dad was a very more earthy person. He had been a lawyer, but then a businessman. But he was kind of a prejudiced person. He, for example, could never understand why I would want to go into medicine. That was just not his thing. And so they were kind of had diverse opinions about what I might do, and of course, me being me, didn't pay too much attention to what they wanted me to do, and I ended up doing things that were fun. So I started in physiology at the University of Toronto in 1947. That was an interesting program because they had a physiology and biophysics undergraduate program, which is not usual, and an honor science program that was really neat. So by the end of my second year, I was starting research in physiology in actually Charlie Best's lab, which has been taken over by Reg Hayes, who was then the department chairman of physiology in Toronto. So that was kind of interesting to me because my aunt had worked with Charlie Best in 1921 when he was developing insulin with Frederick Banting. And so there I was in the same old lab, but doing different things. And so I started off doing the effect of insulin on glycogen formation in rat diaphragm. And the technique was you dip the piece of rat diaphragm into this insulin solution for 10 seconds, take it out, and then put it in a Warburg apparatus and measure the carbon dioxide so that you know what its metabolic rate was. And at the end of the experiment, you measure the amount of glycogen formed. So that was this ancient metabolic measuring device from Warburg from the late 1800s in Charlie Best's lab. So it was kind of fun because it was like doing a historic experiment in two different ways, with a relatively modern ingredient and making modern, then modern, 1948, measurements of the biochemistry, and so that went on. The following year, in the same lab, I was doing experiments on creating giant rats. It seemed it was determining the effect of growth hormone on rat size and the combination of growth hormone with insulin. This, after all, was an insulin lab. So to do that, we got all the beef pituitaries from the abattoir, and then tried to isolate the growth hormone from the beef pituitaries, then injected into the rats. Well, this stuff was not too good. It wasn't really pure, but it worked. And so we would get 400 and 500 and 600 gram rats pretty quickly, the combination of some growth hormone and insulin. They weren't really well characterized in terms of what happened to their blood sugars and so on, but they really had a high consumption rate and really grew well. So that was kind of my start in the experimental physiology. And that went on as I finished that program at the undergraduate level, and then started in other laboratories. So I was in the Air Force Reserve during the summers, and my job there was in the Institute of Aviation Medicine. So starting with Wilbur Frank, I didn't work on the G-suit. That was long done. That was wartime. But we worked on a method for detecting the state of children with burns. Burn is a high stress situation, and it depletes one of the corticoids in the blood in a stress situation. So the idea was to get a measure indirectly of corticoid levels, for which we didn't have fancy biochemical techniques at that time. So the method was to inject a small amount of fluorescein into the skin and then measure its rate of disappearance as it spread out. The spreading is influenced by the hyaluronidase, which is normally in there, an enzyme that breaks down hyaluronic acid in the skin. So the cortisol prevents hyaluronidase action. So that was the kind of crude method of getting an assay technique for the corticoid status of the patient. So that actually, we worked on that for 2 years. Well, as part-time job, and so I would be in the Sick Children's Hospital at Toronto in the nights because you needed to do this when it was dark and so on. That was part of my job as so-called Air Force officer training. And so that was at the Institute of Aviation Medicine in the summertime, being a proper officer after going through boot camp and all this stuff. But then after that job, my next one in the research area was, by that time I was in medical school, was at Defense Research Medical Labs, and where I worked on nerve gases. And I learned how to take out the adrenals from a mouse very quickly. And so that is to say, make a little slit in the skin of the back of the mouse, and with a swab, you take out one adrenal and then the other, and you put one stitch in to seal it up, and it's all done in 30 seconds. And so the mice do fine. And then the test was, what's the effect of adrenalectomy in response to nerve gases? So that was, part of a long experiment, then extended over another year. So I had nice experiences in these labs during my early years, and I was running track at the same time. I ran track for 8 years. While I was undergraduate and then in medical school. So that took time too. But the overall program was really fun. I mean, an MD program is fun if you don't try to memorize everything. And there were parts of it that were nice, logical, deductive things, which I loved. And the physiology research time kept me going at things which had to be thought through carefully and not just memorized. So I really liked that program that I had. Oh, that's a long story, because as I already said, I started with these really relatively simple kinds of experiments, and after I went through medical school, I interned at the Toronto General Hospital, and then I went into practice in Northern Ontario. And so that put me in a very different setting. At one point, I was in practice with my wife's uncle, who had been in this remote practice in the backwoods for 4 years and had no help. He was all by himself. So when I moved up there, it was helpful because then we're two of us for this. We had a 35-bed hospital. I became the chief of surgery, and he was the chief of anesthesia, and I was the chief of obstetrics, and he was the chief of medicine, that kind of thing. So we shared the practice. And, but that gave me a view of how difficult it is to practice integrative medicine, because we did not have a lab that, other than what I could do myself, to do diagnostic testing, and we had to send things off to Toronto, 400 miles south, to get. But the view that I developed during that time was that I wanted to put things together. So I went back into residency training by moving to Hammersmith Hospital and the Postgraduate Medical School of London, in Hammersmith in London, England, for a year. That was a wonderful training experience. They have really good people, and that idea of careful physical diagnosis was prominent in that institution. It was really good. From there, I moved to Mayo Clinic, which was almost accidental. I didn't really know what I was going to, and it was just the best possible thing. It was just lovely for the next stages of my career. So I did a residency there in starting in surgery, but ending up in internal medicine and cardiology. Practiced cardiology and doing research. Well, the Mayo brothers, from their beginning establishment of that institution, had said we should have a three-legged stool of research, education, and practice. And so it was that I went through a program in training in physiology at Mayo and took a PhD between Mayo and the University of Minnesota. So that was a good learning experience too, totally different from the Hammersmith learning experience, but I had fabulous mentors there. I worked with Earl Wood. Earl Wood was the first student of Morris Fisher. Both Earl Wood and Morris Fisher were presidents of APS. And I worked with Vischer in Minneapolis. Well, Vischer was the last postdoc of Starling, Ernest Henry Starling, in 1927. Just before Starling died. So I felt like I'm the great-grandson of Starling through these great intermediaries. So that was the whole combination was a nice experience. Earl Wood was a marvelous person to work with, and gave me all sorts of freedom of the lab to do new experiments that weren't necessarily his and weren't necessarily of his prime interest. But the lab worked as a great gathering of, we had a dozen postdocs in the lab at that time, working very well together on helping each other on different projects. So we all learned a lot, and it was a really good experience in that sense, in a social sense as well as a scientific sense. And the scientific environment was just great because it was one that asked you to learn more deeply, make sure you understand. And the question is, do you understand something that faced us each day as we were trying to figure things out? So that was my residency training, and the PhD, though, was a side issue, but still valuable in the sense of giving me more quantitative aspect of my work. My first APS meeting, I had a paper to present, and there are a lot more papers presented than now, in a sense. And so there I was, a green postdoc, presenting my paper when who walks up to the front of the room but Louis Katz and Alan Burton and these two giants. I'm saying, oh, I'm in trouble. Because Louis Katz was known for asking fierce questions, or asking questions fiercely, I suppose I should say. Alan Burton, likewise, they're very probing people, and they sat together and listened to my talk. And it was okay. They approved and talked to me afterwards. So I had that great sense of relief of getting by on my first presentation. That was Atlantic City. All these first meetings were in those hotels on the boardwalk where you meet everybody. Live in old hotels. We lived in one time up the boardwalk where the windows were painted open and it was snowing. So the boardwalk was great, and we haven't had meetings there since they started gambling in Atlantic City, but well, it's almost as cold here in Boston this week. There's so much. I mean, it's hard. There's so much physiology to learn. You cannot learn it all. And the way I look on this biology, the explosion of molecular biology and genomics, and trying to link it to the integrative physiology that's my heart and soul to work on, is not easy. So from that point of view, I'm thinking that the students need to be able to study, ask themselves at every stage whether they understand not only the individual ingredient item that they might be working on, but do they understand what it's for, what it does, what it means in the context of the whole organism. And that's really hard to do. And as I went along over the years, we put together a concept that I called the physiome, which is the idea that you can develop a quantitative, integrative description of function of an organism, potentially in all its detail, through quantitative mathematical modeling. And so we developed that. Well, I presented the name of the physiome over 20 years ago. And then with the help of the IUPS and their council, we developed it within the International Union as a facet of the physiological congresses through labeling it bioengineering at first, and then set up the Physiome Commission, which is a broader way of handling it within the IUPS. So I've really put my central thinking toward how to develop the Physiome to make it available open source as reproducible research. So that's meant blending the physiological research with the technologies that preserve that research and allowed people to build on it. So creating open source simulation systems, open source models, and now with the development of open source publication, we can start to close the loop on this and have the Physiome presented as sets of mathematical models which can be brought together to help understand the whole organism. So that's what I've been trying to do, and that's what I try to teach the students. Well, it's not easy because the level of knowledge that you have to have in these technologies, like doing simulation and mathematical modeling, is non-trivial. Learning the biology is non-trivial because it's a huge area and growing. The ignorance explosion far exceeds the knowledge explosion. And we learn more and more, especially by doing the mathematical modeling and discovering what you don't know and you would like to know to understand that model. So that's a big development. So it's harder for students to gather all of this at once in a PhD program. So it really necessitates having PhDs that have multiple components, and then also using the postdoc experiences to gain knowledge in other particular areas that will blend and allow you to produce the kind of research you want. One example of how one person tried to develop his program was Joe Serchevsky, who became chair of physiology at Mayo Clinic. But I first met him when he was a research fellow, first-year postdoc with Charlie Code. Charlie Code was an eminent gastroenterologist and worked on gut motility and was president of the American Gastroenterological Association and my chairman. But Joe Serchevsky worked with him on gut motility. Then he took another postdoc with Molly Holman in Australia on acetylcholine receptors. And then he took a third postdoc with Edith Bulbring in Oxford in pharmacology on the electrophysiology of smooth muscle. And then he came back on the staff of Mayo Clinic as a brand new assistant professor, 5 years of postdoc. However, in his first year as an assistant professor, he was invited to give a lecture to the Royal College in Great Britain. He invited to give special lectures at the American Physiological Society, and he got a grant. He was on his way. And the virtue was that he took these 3 different areas of smooth muscle physiology, put them all together. And then people had not been trained as broadly and deeply as he was trained in gastroenterology at that time. So it immediately made him a prominent expert by virtue of carefully choosing the right kind of training. So that's just kind of an aside, but watching Joe Serchevsky do that so successfully was rather nice. I have this theory that I shouldn't even call it a theory, I should call it a strategy. And it's to look at physiology from the cell point of view. When you think about it, the regulatory things that occur are occurring at the cell level. You express signals from maybe an external source, or maybe from inside the cell as you change metabolism or something. But the response is, when you change the cell function, is to change the signals for transcription. So those come downward from cellular function to the genome. You transcribe certain kinds of selected groups of proteins. So you take them out of the recipe book and create the new broth that is selective for the purpose. So that's driving from the cell down. There are a lot of valiant attempts to discover from the genome up, and some of those where you have a single gene disease are very effective. On the other hand, from the point of view of discovering how to regulate biochemical networks, it's not so effective. And if you do mRNA arrays and do association studies, then you try to map pathways of expression, but the information is not usually gathered with enough resolution or over enough sequential points in time to really get much information that way. Likewise with protein-protein interaction networks, that's really hard to do. It's difficult. And they take information on yeast 2 hybrid, propinquity on the genome, the near neighbors of genes are likely to have like functions or related functions. You can look at phylogenetic heritage, and you develop these PPI networks through massive computation. As of 3 years ago, there have been 50 of those developed, and none had been validated. Oh, well, how come? It's partly the social structure that the computational science guys who work out PPI networks using good mathematical Boolean algebra techniques and so on aren't the people who are going to do the experiments to test the network. And they're hard to test because it's hard to know which way to go around to do the experiment to test what is now a big network. So the alternative view that I'm recommending to students is look at the cell. The cell is providing the information to go upward to structure and function of the organ. And it's perfectly correct that there are humoral and neural signals which come down to the cell and evoke responses. But the central point of effectiveness is through cellular changes in cellular behavior. So work there and work both ways. And I think the fastest way to get to uncovering the genomic and which genes function in which ways is to track down from changes in cell transcription and make the observations there to try to find out all the little proteins, the helper proteins and the insertion proteins, and the ones that really make transporters or enzymes effective. The ones that you know about are being helped by other proteins or being moved along tracks into the cell or out of the cell. And so but starting at the cell level is going to be really useful, I think.